So this morning section will have our first speaker is Professor Lucas Bleicher. He is from the Universidade Federal de Minas Gerais, and he's going to talk about coevolution analysis and some bioinformatics. And so, Lucas, thank you very much for coming.
Thank you for inviting me. Well, I'm glad that a couple of days ago there was a presentation from a postdoc talking a little bit about coevolution as well, so it will save me some time explaining stuff. So we're developing this method to get information on protein families. So, well, we all know that when we are studying new proteins, there are many new things to discover, and also we all know that due to all those genome projects, there's increasing number of new sequences being deposited in the data banks, and there's no chance we can study experimentally or even in silico of them. And so what usually happens is that once there's a new genome project, there's some sort of automatic annotation, so use some softwares that will at least give some clue about what may be the possible functions of these new sequences. And given the case in which there are at least some homologs which were previously studied, so this makes this task a little bit more simple, and this is where we start. So one thing most people know is that sometimes it's much more useful to try to find new information on a new protein, not only looking at a single sequence, but looking at its context. So protein families, or at least most new proteins are embedded in protein families on which we can find a big number of homologs. And the first thing that one can do, and it's quite trivial to do, is simply look at a set of homologs and see which are the positions on which all sequences or virtually all sequences have the same residue. So if this happens, there must be a good reason for that, and that's the first clue we have about finding functional residues, things that are on active sites and things like that. So this, for example, is something that was published in the 90s. This guy was studying this protein, which was a laminar nase from Rhodothermus marinus, and there was no structure by that time. But he knew what was the reaction that was catalyzed by this enzyme, and he knew all other examples of proteins which are homologs and catalyze the same reaction. So simply by doing a trivial sequence alignments, he noticed that there were three positions where all sequences had either an aspartate or a glutamate, and those are residues which are commonly involved in catalysis. And so he did a site-directed mutagenesis on these residues, and the protein ceased to have activity. So it's one kind of example in which you can go directly to which has more chance of finding which are the functional residues. So this shows that experimental or computational studies can be guided by something quite easy to do, which is simple consensus analysis, and this is something that most people do already. What we set out to do was to go beyond consensus and look at coevolution. What we see on most protein families is that in many cases there is not only conservation, a given position which most sequences have a given residue, but also coevolution. So sometimes we see that certain positions, when they have a given residue, it's quite probably that another position will have another one. This is nothing new. This has been discussed since the 70s, and by the time it was focused on the compensatory mutations, which Ricardo discussed a couple of days ago. By the time there was not much availability of protein sequences, but as we said, this is going increasingly higher over years. And so in the 90s, there was an important article from Gebel on which he proposed that if compensatory mutations yield coevolution signals when you look at a multiple sequence alignment, then we could do it in the other direction and detect such signals in order to predict contacts. So this was suggested in 1994, but by that time he already noticed that it was not quite efficient because there was a big rate of false positives. So this being the case of distance residues in the structure which co-evolved as well. So this was a bit dormant for some years. Then in the late 90s there was a group led by Ranganathan who proposed a new interpretation on why there were those co-evolved sets of positions in protein families. And he proposed that when one found a set of residues which appear simultaneously, they could be related to a loss theory, and that those coevolution signals could be used to predict delta G values for mutation studies. That was something that sounded too good to be true, and unfortunately it wasn't really true. So after some further tests, the people noticed that if you tried bigger number of mutations, there wasn't this correlation between delta G and coevolution which he was proposing, and there was not a physical basis for trying to relate coevolution with a loss theory. So this was rebooted in later articles. But by the time he was proposing this method, I was working in São Carlos, and I was trying to study a protein family, which is the iron and manganese superoxide mutase, which was discussed yesterday. And we decided to use the method he proposed, which, although the initial interpretation of the results was not really true, but people found that at least it was good enough to predict sets of residues which were indeed important for protein function. And so we decided to study this protein family with this method, and what you see that heat map is how much the distribution of residues in a given position changes when you look at what is in a specific position, which is different from that one. So for example, if you look here in aspartate 170 and position 72, it's quite reddish, which means that there's a given residue distribution in position 72, and this changes a lot for the sequences which have an aspartate in position 170. And if you look at one of its more bluish, for example, T24 and 145, it's blue because there's a given distribution of residues in position 145, but the fact that you have a 309 in position 24 simply doesn't change what you see in this position. So this is a way of finding coevolution. And so when I was working with this with Richard Garrett and Fernando Bachega in São Carlos, since they were quite familiar with this protein family, by the time they looked at this result, on which we see three different sets of positions which seem to be simultaneously co-evolved, they looked at it and say, oh, but this is exactly what was already described as positions which are typical found in the superoxide mutases that use iron, which are on the top right, or those which use manganese on the bottom, and also those which determine if the protein is dimeric or tetrameric, which the two options exist in this protein family. And those that were previously discussed are these which are underlined. But the nice thing is that we found other positions using this method which were not described before. And the method that was used to find this position was based on training. So you had a set of proteins which people already knew the answer if they used manganese or iron, if they were dimeric or tetrameric. And then the method simply used these results to train an algorithm and apply it to a test set to see if they were able to predict what would be the bound metal or the oligomeric state. And what we found out is that we could also find, well, these positions in other ones which were just as good to predict the bound metal or the oligomeric state without imposing any information about what we are supposed to find. So This means that, and this is what we use as our own interpretation of coevolution in protein families, was that if we know that a given protein family has subclasses, so for example, some of the proteins in the family bind DNA, others don't, or some do this reaction, others do a sort of different reaction, some have a region which interacts with membrane, others not, things like that. So probably it won't be just one residue that will determine if this happens or not. Probably it will be a set of them. And this means that if the multiple sequence alignment is sufficiently populated by proteins from different classes, then we are expected to find these sets of correlated positions by doing an analysis like this. So this was made us quite optimistic about the results, and then by looking at the literature and seeing discussion about this, we found out that for things that most people agreed with, there are at least 3 known reasons for finding coevolution signals. The first one, then it was the first that was discussed, and Ricardo a couple of days ago already mentioned, is the structural contacts. If residues are in contact, we are expected to detect that what we have in this position will probably affect what we have in this position. The other one is the phylogenetic effect, which isn't something that we can use for studies. Exactly, it's sort of noise which we must correct. And the third one, which is what we aim at doing, is detecting functional correlation. So in this case, in which there are subclasses which do somewhat different things in the same protein family. So if we detail each one of them, as for structural contacts, Ricardo already discussed this a couple of days ago. As I said, in the 90s, people already were trying to find methods that could use coevolution signals to detect contacts, but by that time they weren't really successful or even useful. But one really nice thing is that nowadays we have methods which are good enough to predict protein contacts which are so reliable that they can even be used to predict folding. So there is DCA, which Ricardo discussed it, and PsyCov, which is another method which uses this information to find contacts. As for the phylogenetic effect, this is something that relates to the history, the evolutionary history of the protein family. So using a simple example, if there's a set of proteins which were generated by this ancestral which has A, R, and D in a part of the sequence, and then there was this speciation event here, and the last residue was mutated to E, and in this branch the first was mutated to C, and then there were a lot of speciation events after this mutation, and given the what we have today in the genomic databanks, we have a lot of sequence from all these species. Then what happens is simply that we will have sort of correlation, detect a correlation between having an alanine here and a glutamate here, and having a cysteine here and a aspartate here. So it may be not because there's some functional importance of having those residues simultaneously, or because they are in contact, but simply a result of what's the evolutionary history of the protein family, and most importantly, how the sequences that we obtain were treated for example, redundancy and things like that. So this is actually something that unless we are really trying to study how the protein family evolved, it's a sort of noise in our data. We have methods to try to get rid of those signals, because for what we are looking for, they don't add anything useful. So, and the last thing is functional coevolution, which is what we decided to focus. So there are other groups which are already doing a great job in detecting contacts, so we decided to go the other way and find and propose a method that would find functional coevolution from what we have in protein families. So in this case, as we showed, if there are subclasses in a protein family, then we can find the sets of residues which are related to this function. And the way we explore this property is doing things that were not done in other methods. So for example, most methods, they just focus on looking at what is the signal between two different positions. But for function, the identity of the residue is important. So we know that some residues may be related to given functions, others don't. So the method is specifically based on residue identity and position. Also, there are cases in which anti-correlation can provide useful information. So sometimes when you see that having this residue means that we don't have this other one in the other position, it's something that can be useful for us. For example, when we have classes which are mutually exclusive. So sometimes in the same protein family, the fact that you have this characteristic means you won't have this another one. And also, it is desirable that the method to detect the sets is not susceptible to subjective or visual criteria. So, for example, in this method I discussed it before, what we do is basically calculate a matrix for all positions which have sufficient which are sufficiently populated, and then we get rid of low signal positions, and then through visually inspect to find the sets, which, as you can probably see, is something that is a little bit subjective. I gave the same set to 5 different persons. It's not unlikely that I have 5 different results. So what's the method we proposed? We call it the composition of residue correlation networks. The first thing we do is to weight pairwise coevolution using residue-specific criteria. So, for example, in this position, I have 67% of aspartates, and for the sequences which have an arginine in this position, this goes to 100%. Of course, this is, if we calculate what's the probability of such thing happening, it won't be quite unlikely because it's a 6 sequence, multiple sequence alignment, but the kind of alignment we use at least some hundreds, in most cases a few thousands of sequences, and so we will find that in the cases which are really co-evolved, the probability will be really low. And so we define a coevolution score as minus the logarithm of this probability, because this we can have quite a big range of values for p. And if there is anti-correlation, so if you have this residue, I won't have this one, so I just revert the signal. Then I build a network with these results on which having a node means that I have a combination of position residue. So for example, glutamate in position 18, and I will have a connection which is weighted according to the score. In order to have something useful, I just use nodes and connections which are above a given threshold. Then we can have things like this. These are idealized examples, but so we can think of cases on which such networks would happen. So for example, if we have a protein family on which most proteins are enzymes, but there's a branch on which they lost this function, then we can expect something like A, on which the residues involved in catalysis tend to appear simultaneously, but once you lose one of 2 of those important ones, there won't be evolutionary pressure to keep the other ones, and so there will be just this network. If I have a protein family on which there are 2 independent characteristics which are represented there, for example, in the case I showed the fact that a protein uses ion on manganese on superoxide dismutase, or they are dimeric or tetameric, are not related to one another, so I can have sets which are quite correlated between them, but there's no correlation, there's no cross correlation between these sets. And finally, if I have a given characteristic which is mutually exclusive when compared to another one, we can expect something like this. The red lines means there's anti-correlation. So for example, if we have a protein family on which all of them do a given a certain reaction, but they can use 2 different substrates. So for example, one does a reaction, but the rest of the molecule is quite hydrophobic. The rest is the same reaction, but the rest of the molecule is hydrophilic. So we expect that since it happens on the same site, there will be a lot of residues which must be simultaneously there to detect the hydrophobic substrate, and we must change all of them to detect the other one, and so we'll probably have something like that. Well, sometimes networks can be quite complicated. It can be things like this or even like this. But thankfully, there are all of our friends in physics and mathematics which are working on methods to decompose networks. Basically, what we do is to you have a quantity which is called modularity, which basically says how well are you dividing a network in subsets, and this depends on each set is quite connected with the members of the same set and is not really connected to the rest of the network. We can calculate a number to find this, and applying this to the case of superoxide dismutase, we have this network, which is quite informative. So for example, these residues are those which are related to having manganese in the binding site. So we see that they are quite connected, and there's anti-correlation with this other set, which are the residues which are found in ion binding sods. And there's also this set of residues which is found on dimeric sods, and as we can see, the relation between the sets is what we would expect for the function. So in most cases, the protein either function with iron or manganese, and not both, and so we have a negative relation between the two sets. But being dimeric or tetrameric is not really related to what metal you have in the binding site, so this set is disconnected from the rest. We also found two positions which are in contact. This is a couple of phenylalanines. You see the difference here is 4 because they are in the same helix and they form a pi-stack interaction. One interesting thing we also did was to develop an adherence value which says for a given sequence how much they fit in this coevolved sets that we detect in the protein family. And so we see, for example, that ion sods, they have a high score of adherence to the set 3, which is the one here, and which is we found out already that were related to having iron. Manganese, it would be the other way around, as expected. But an interesting example for which we see in C is that there's a small set of superoxide dismutases which are called cambialistic that they can do the reaction both with iron or manganese. So they accept both metals. And what we see is that we have sort of intermediate results of adherence. So this was something that was quite exciting for us. And this is for the oligomeric state. The dimeric sods, they will have positive values of adherence to the set that was on the left. The tetrameric won't have any of them. So we apply this to other systems. So Marcelo, which is in the audience, did an analysis on nuclear receptors. We found some interesting things on a divergent set of proteins in this family on nematodes. This is something we did with a protein from yeast. This is from a group in São Paulo with which we collaborate a lot. And this is the one we published last year, which is also a yeast protein that was uncharacterized, and they did a lot of mutagenesis experiment using the positions we found using this analysis to see how they would affect the phenotype of the Saccharomyces cerevisiae. So I'll choose the one in the middle, which is, I think, is quite interesting. What we see here is the final network, but I have already cleaned negative correlations and also those that were left between different sets, so that it would be easier to look. Otherwise, it would look as ugly as those I use as example of complex networks. But they were studying this protein HER2P, and this is belongs to a protein family which has a lot of different functions, even though all of them are homologs. So we see 3 different types of independent enzymes in this protein family, and there's also some of them which they do not catalyze a given reaction, but they actually are part of a complex which is called the transamidosome. So since they were studying this specific this specific protein in yeast, what we found out is that one of the residues, which is the most interconnected one in the left, when we looked at that in the structure, we saw all those residues were in the same side, and when we compared this with already known structures from the transamidosome, we found out that they were exactly on the interface which would connect the protein with the rest of the complex. And also the proteins which did have these residues were those that were described as part of the transamidosome, and so did the protein that we're studying. So we use this as a case to propose that the protein from yeast would form such complex. They did some cellular localization experiments, and so that's what we proposed in the article, and we were quite lucky because just a couple of months later there was a published structure from this protein which confirmed that it was indeed part of a transamidosome in yeast. So finally, as for an ongoing project, we are currently studying this family of transthyretins and HIUAs. Transthyretins are hormone carriers. They carry the thyroid hormone and also can transport retinol in blood. These have this tunnel which is formed when the protein is in this tetrameric form. And a few years ago, people found out that these proteins actually have distant homologs in other species on which there would be no reason for them to be doing the same thing. So we could find these in bacteria and things like that, and you wouldn't expect bacteria to need a protein to transport thyroid hormones. So they were called transthyretin-like proteins, and it was found that what those homologs actually do is a reaction. So they are part of uric acid pathway. They do the second reaction in the pathway. And as for the evolutionary origins, there was this gene that is quite old, as I said. It's older than the separation between prokaryotes and eukaryotes. And there was a gene duplication in early vertebrates which resulted in a second protein, and that's what evolved to modern transthyretins as the one we have today. The interesting thing is that the original gene, the one that did the reaction, was inactivated in humans and chimpanzees. So this happened because that pathway had already lost the protein that did the first reaction, so there was no evolutionary pressure to keep the second one, so it became a pseudogene. And when we did this analysis, what was found, well, what we would trying to find was, well, since there are two really different functions using the same structure and the same kind of protein, what would determine if a protein will do the reaction or that will carry a hormone? So the first thing we did was a simple analysis as those that I showed. That the only thing we found out was a set of residues on which most of them we will see is consecutive numbers. So this is the C terminal of the protein for HIUAs, which is the one we don't have, and also an aspartate which is in the exit of the tunnel, and also a salt bridge. So sometimes we also capture structural pairs using our method. So our hypothesis for our method was that if there's a protein family which has subclasses and these characteristics are determined by sets of residues, then these determinants should naturally arise from a correlation analysis if the alignment is sufficiently populated by these classes. The problem here is sufficiently populated by these classes. As I said, the transthyretin are really, really new. They appear in vertebrates. So even though this protein family is present in more than 6,000 species, transthyretin appeared after duplication, which happened in early vertebrate. So we have this quite big discrepancy between the sampling of the two kinds of proteins. So sequences for core data are less than 5% of the total number of sequences. So how can we deal with that? The first thing, we kind of cheated because since we already knew where the substrate and the hormone binds, we just calculated what would be the correlation for these positions alone. So these which will be in contact either with the substrate or the hormone. And then those which are in green means that this position is present in those proteins. So those which are here are basically HIUases, and the rest are transthyretines. We can see a pattern here. There's a given position which is present in all of them, but this is something that was already described as important actually for the formation of the tetramer, not the function that the protein can have. And if you see the positions which are related to binding hormone, you also see that it's kind of the other way around, although there are some positions which do not show this same pattern. How can we do this without cheating? So another approach we did was to, as I said, taking a branch from the tree of life, which is basically in order to have a better sampling from the two classes, we started with core data sequences only. Well, this has a bad thing that we'll have poor statistics, but at least we have a better balance between the classes. So it's much less sequence. We have only 72 after removing redundancy, but we still get some signals. And we use the same protocol, but we used a smaller threshold to build the network. And what we found is that there's quite a big set of positions, but they are two, which is what we expected, and we hope they can be related to what makes HIUAs different from transthyretines. So this is an ongoing project. When you see what would be the HIUA specific residues, they are mostly concentrated in the central region, which is where the function happens. For transthyretin, they are everywhere, they're all over the place, and this is something that worries us a little bit because there is not only, or the function of transthyretins is not only carrier hormone, but they also have some zinc sites which are distant from the tunnel, so things can get a little complicated. But so what we are setting out to do first would be do molecular docking. So there's no known structure from HIUAs with its substrate or the product, although there are a few for close analogous. But so we will dock the substrate in the product, and also the reaction intermediate to the active site, and then we can have a more reliable picture of what are the residues involved within this reaction. And there's also something we hope to simulate in the future. And sort of our dream project would be using this as a proof for protein engineering, which is the idea to see if we can interconvert HIUAs to transthyretins or vice versa. So this would mean that if we can detect which are the positions which are related to each function, so if we mutate one for the other, we could check if this actually is the case. And this is something we hope to do in the near future. So the conclusion is that this method can be used for data mining. We have articles on which we basically reviewed protein families, looking at which positions are important for function, and sometimes there are things which escape the analysis of other groups. We can detect positions which are putatively involved in functional subclasses, and this can be really used for designing side-direction mutagenesis studies. So this is what we do with a group of Mário Barros in São Paulo. And they can also help guide simulation-based studies. So since we find positions which are putatively involved in function, we can see if that's really the case by using simulations. And what is quite interesting is sometimes we can try positions which weren't previously discussed, which people didn't notice before. And the great thing is that a complete analysis is computationally very cheap. I also say that usually we take an afternoon to generate the results and then a month to interpret them. But even this is getting easier because we now building our own software, which is called PFStats, which is, well, we already wrote this before, but now we are doing it in a way that other people, well, other than me, can use the method. And so we have this the software which can you can use your own alignments, or you can download them directly from PFAN. It can generate the networks, and you can see the results on screen. It shows how the positions you found are distributed on sequence you choose as reference sequences. So that will help to compare with those that were already studied. It can show also the results, so in structures. So this, for example, is protein which is colored according to conservation, and so the positions which are in red are those which most proteins in the family which have basically the same residue, or not a big difference between them. And finally, something that's helping us quite a lot is that we are also directly obtaining information from UniProt. So once we find the positions which are conserved or which are correlated, it automatically looks for all proteins in that protein family. If there is at least one article or one reference or one entry in UniProt which says, oh, this position has been already discussed this because it does that or that. So for example, we have some positions which were described as being part of an active site, or binding a given metal, or forming disulfide bonds and things like that. Oh, by the way, beta testers are welcome. We hope to publish this year, but we're already quite advanced in doing this. So I'd like to thank CAPIS, NPQ, FAPMIG, and UFMG for all the support. I also like to say there's well, this is an invitation for an upcoming event. So the annual congress or the computational biology society in Brazil will be in Belo Horizonte this year in November. So 16 to 18 November. And this is our group in Minas Gerais. So we also have in our group Rafaela Ferreira, some of you may know, which works with drug design, Doc methods. Lisa Felicore, she does synthetic biology, and Ronaldo Najin is our crystallographer. And that's it. Thanks a lot for.
Thank you, Lucas. The floor is open for questions. Sergio,
this was very nice. Just about the method, two questions. First, how do you deal with gaps in the alignment? What does produce in your correlation? And second, or if you want to answer that, and
well, as for the gaps, gaps are considered, but given that positions which are gap-rich are usually those in which the alignment is poorer, then the result is that they automatically yield low scores. So we do consider gaps, but that will mean that if let me get back to how we calculate this. So what we do is, the first thing we do to calculate the pairwise correlation is we see for a given position which has a given type of residue, we calculate its frequency and see how the frequency varies for another position with that distribution. If a position is gap-rich, this means that the number of sequences you have would be lower. And since we use a binomial distribution, this is sensitive to the total number you have, and this will get automatically lower values for actually for the score. And so you're not expected to see high correlation values for gap-rich regions unless they are really strong. For example, if you have a position which half of them are gaps and half of them are a given residue which is correlated to another position almost exactly.
And the second one is, can you combine this with the other data mining information that you have? I mean, if it is a viral protein infectivity, or if it is
Different proteins.
Lipase, or an enzyme, the activity, or stuff like that.
You mean?
I mean, once you have your correlation matrix, and then you identify the points that are correlated, can that help if you can? I mean, can you cross-check with information about, suppose if it is an enzyme with the activity that you can get in the literature? Is it possible to increase in some way in the level?
We don't do this automatically. This is something that, well, in some cases we actually do this, but it's a manual process which takes quite a lot. But this is something we definitely want to do automatically. So the guy who's writing the program, he actually is an enthusiast of data mining method. So we'll probably do that at some point. But up to now, we're kind of doing manually. We find the positions, we look at the literature to see what are they related to, and then we do the conclusions. But it's not something that can be automatically done at the moment.
It could be a way to filter out false positives, right? I mean, if you could combine with cross the data with data mining and with results from literature about the kind of high throughput, that would, I guess, be helpful to filter out the false positives or
yeah.
pinpoint really points that are really related with something.
Yeah, something can be done. The thing that it's difficult for us in the moment is that many times it's hard to find residue-specific information from things we actually can mine. So we do this for UniProt sequences. Some of them are annotated, but the best thing we can do, and unfortunately we still do this manually, is using Google Scholar. So the nice thing about Google Scholar is that it actually looks inside articles, which even those which you don't have access to. So you can, so for example, you have this position we found for a given protein, and that we do a Google Scholar search and see that in some article people describe that this position is related to that. But as it seems that Google forbids using Google Scholar for robots. And the only thing we can do at the moment, and this is something we aim to do, is using the database from PubMed, because at least they release the abstracts. But most mutational studies don't list all the things in the abstract, and it's quite sad, because if we could do this automatically using Google Scholar, it would be a huge improvement in the method. So we sort of do it manually at the moment.
This is because most of this information, it's not in a easy way of accessing. So it's it's
so
like a lack of a good policy on data distribution.
Yeah, well, because see, for example, if you have an article from a crystallographer, most of them do a really good job explaining which positions are important for functions, but there is something that is buried inside the article. So we will find, aspartate position 180 is important for doing this, but this is something that usually is not in the abstract. And we can find this information using Google Scholar, but it's something that we need to do manually. And I don't know an easy solution for doing this.
Sci-How?
Oh yeah, that can be a good idea.
Why did you exclude the copper zinc salt when you study the manganese?
Okay. That's because the copper binding salt is actually a different protein family, so they don't align with iron manganese salt.
Just to have an idea, how many proteins do you actually need to have a statistically significant result?
So the answer is exactly how many we need after we do the filtering, because sometimes we have families and which have a huge number of proteins, but once we remove redundancy, we have just a few. And so after we do a redundancy filter, when we really eliminate those which are quite close to each other, the ideal number is at least a few hundreds. So the example I showed when I used just coordinate sequence, we had only 72, which means it's quite noisy. So it's not the ideal case. But with a few hundreds after I do the filtering, it's okay.
Second question, what is exactly that you call redundancy? Because I imagine two proteins of the same family that could be very similar, would that be considered redundancy?
Yeah, so for example, most of the time I eliminate, if I have a couple of sequences which are higher than 80% identity, I remove one of them from the set. And if by the end I have a few hundreds, then it's perfectly okay. And that's usually the case for most protein families, unless it's something that's really recent in evolution.
And can you use that to trace evolution back to see the
Yeah, this is something we are implementing at the moment. So we are working with people from evolutionary biology, for example, for mapping how these residue distributions are given a phylogenetic tree. Again, this is something we're not doing automatically yet, but it can be done.
Although you talk about the evolutionary correlations that might be driving coevolution, I'm not sure if you use it as a parameter in your model, or you try to consider it anyway to model the probabilities of coevolution among sites.
No. So this is something.
There are many evolutionary comparative models that can reveal that.
Mm-hmm.
So it's not too hard.
Well, I'm glad you say so, because that is something that we were quite a bit afraid of doing. Because, well, we know that there are quite sophisticated schemes for doing this. We actually use a really simple one, as I said, removing proteins which are quite close. But, well, if you say it's easy to do, I really let you
What
try.
about the chemical properties of this substitution? You can deal with that in your model?
Well, this is something that we already implemented, but we didn't do some test cases to check. But for example, we can do alphabetic reductions on which we try to assess with the same. So we already have the routines that do that, but we just didn't do a big test to see how this would help our case. But it can already be done.
Okay, so we should move on. So let's thank